KOIOS is a tool. It's kind of the brainchild of the oncology genomics work group, supported by the Roux Institute at Northeastern and Odysseus Data Services, of course. KOIOS is designed to kind of solve a problem in observational precision oncology, which is that to do epidemiology in precision oncology, we require a set of very concise, very standardized, and searchable interpretations of genomic variants, of which there are potentially trillions. And we need to do this so that we can apply the epidemiological methods that we would want to in OMOP to clinical research in genomic variation. And this is difficult on the surface because variants are not concise, standardized, or very searchable. And genomic variant data is somehow even worse than the reality. It is not a standardized set of data sets, is not very interoperable, and this kind of data can come from a huge range of sources, all the way from whole genome sequencing, RNA-seq, down to immunohistochemistry, to a personal doctor note identification. So we have a combination of very, very narrow, very measured sources of data, and then almost wholly meaningless sources of data where the thing that we're interested in is very sparse in next generation sequencing. These come from different methods, they come from different modalities, and each of these modalities has their own world of representation, which can be more or less meaningful. This is just a screenshot of a VCF file. That's a whole genome sequencing file that is used to track variants. And there's a lot of information here. Almost all of it is useless to us, or at least not particularly interesting. But what we want is a set of features. We want to be able to say that this patient has this feature or these features 1, 2, and 3, and they don't have feature 4, 5, and 6, or at least we don't know if they have feature 4 or 5 or 6. And that is certainly not what we have as we've just gone over. So when it comes time to ETL your genomic data, especially if you have whole genome sequencing data, it's easier said than done. As I say, we're pulled between this idea where we have different modalities, different sources, different methods, but we really want precise, standardized data to be able to do this epidemiology. And what we have are flat files with complex syntax requirements, really complex syntax requirements, but astonishingly a very, very lax compliance in sticking to that complex syntax. And another thing that we really need here is a closed world assumption. There are kind of limitless genetic variants, or at least too many to count. But really, we want to be able to define a closed set that are clinically relevant or useful. And to do that, we need to be able to ingest the data in the first place. So it's a very high level overview again of the process of KOIOS, but essentially all we're doing here is I'm trying to explain how we pass a VCF file. We convert it into a format which is much more standardized in genomics called the HGVSG. And we are able to use this to kind of assign an identifier to each variant. We then use an external resource called ClinGen, which is kind of a global database of genetic variants, which contains a lot of the linking information that we'll use in the genomic vocabulary hierarchy. But importantly, it contains certain strings which we can associate with the concepts that we have in Athena that already exist in the OMOP genomic vocabulary. Very brief overview of the kind of algorithm that we use to create this HGVSG code. I won't go into it, but we can talk about it if you ask. It's really very, very simple. So quick software demo. I hope you can still see that, and it will be very, very quick. It's an R package. You can download it now from GitHub. The link will be at the end. All we have to do to run KOIOS is load a concept set. This is just downloaded directly from Athena and then put into the package as a data file. We want to load our VCF. We want to set our reference because it's much quicker, but we can automatically detect the reference of the VCF. And here, this is just a setting that is not very interesting. So we select that. We have a little pipeline at the bottom there called multi-VCF pipeline. We have about 5 VCFs in the folder that we're referring to here. And what we're interested in is going to come out of this. We got about 10 to 15 records in each file, so we're just processing through those. Sometimes you might have thousands of files. Sometimes you might have one file which contains thousands of patients, but KOIOS should be able to handle it. And ta-da, this is your output from the kind of VCF file that you saw in the presentation. Very, very complicated, but here we have a very concise format. We have a list of concept IDs. We have a list of genes that are affected by the variants that were in your files. We have their concept names, their community titles, all of the different kind of identifiers that you might be interested in, and information on the downstream protein effect so that you would be easily able to search that. At the moment, KOIOS is kind of a tool that's hopefully going to be used in ETL, but eventually we hope to use this tool in reconstructing or at least bettering the existing genomic vocabulary using the hierarchy from ClinGen. And hopefully that was quick enough. If there are any questions, I'd love to hear them. Oh, actually, let me share one more thing. Sorry, let me share one more thing very quickly. We would love to have you at the Oncology Genomic Work Group, which is every second and fourth Tuesday at 9 AM ET. Here's where you can download KOIOS. Special thanks to Asia, Christian, and all of the rest of the members of the Oncology Work Group. And special thanks to John and Fanny K. Shaw for access to developmental data that we used to make it. So, any questions?
Great, thank you so much, Laurence. Oh, we do have a few minutes here for questions before we go to Andre. Any questions?
I'll assume you're saving them all for the work group that you'll be coming to next week.
We do have one from Andy.
Wow, it's first of all, wow. I did take, or there was a workshop at the recent AMIA meeting, and there were several conversations and presentations around fire and capturing this information. There seemed to be a fair amount of challenging pulling the right information out of the report to codify. And then because of the different ways in which information is codified in different terminology and specificity, operating on it and actually deriving values was challenging. It seems to me that you're leveraging Athena's relationships to try to overcome some of those challenges. Did you find gaps in Athena, or like did you have to make a lot of subjective choices in the code?
No, in a way there is an existing hierarchy in Athena. In a way we're kind of bypassing that, although we do hope to use KOIOS to maybe develop on it in the future, because as you said, it's very complicated and it's not yet complete. We rely on the hierarchy that's within ClinGen. ClinGen itself reports all of the downstream transcripts and proteins. And so if you have a protein or you know which protein is affected, you can go up or down that hierarchy to the genomic variant. But the point is that hierarchy should be usable so that if you have a doctor who says this patient has a known very common defect in this gene and this VCF file contains that on line 700,015, then you can combine those two pieces of information using KOIOS. But yeah, it's tough, and hopefully the hierarchy will be perfect one day. But yes, there are some gaps at the moment, as there are guaranteed to be.
Thanks.